Bill Nye the Science Guy. Bill Nye the Science Guy.
Science rules.
Bill Nye the Science Guy.
Inertia is a property of matter. T minus 7 seconds.
Bill Nye the Science Guy.
Hey guys, JJ the Science Guy here. Today we'll be talking about protein synthesis. It starts with DNA. It's what makes you, well, you. First, we gotta know there's a template side and a non-template side. The template side is the part we copy, where the non-template side, it's there for, well, support. So the first thing it does, it opens up and stretches out like this with an RNA polymerase. What it does is it gets into little cracks and crevices, and it opens up into this weird positional wall, and it turns into RNA. But this RNA, it's not filtered. It has to be purified.
Okay.
And for that, we'll have to begin to my sidekick. Oh, you know? Okay, got it. So, this is RNA right here, this long strand, and it's filled with exons and introns. The exons are the part you want, the part that's really good, the stuff that makes protein. But the introns are just the filler to make it long and elongated like a strand. But first, we have to look. There's pieces missing. And these pieces, what makes the poly A tail, which goes onto the 3' side, and a methyl cap, CH3, to go to the 5' side. This is ready for our next step. Perfect. Now that we took off all the introns, this thing is ready to go to out of the nucleus. Hey Bill, catch. Oh, come on. Now we have to put it all back together.
A few inches later.
Now that our mRNA is purified, it can go through the nucleus, and now it can meet our ribosome. This is our ribosome. It's comprised of two large units made of rRNA. This is a large subunit and a small subunit. Together, they clamp like this, and it makes an R site for our mRNA to go through. But before that, we must know the three main primary sites, E, P, and A. These are where the tRNA stand. The P site is where the tRNA goes. So first it's going to go through like this. Through like this.
Okay.
Into the RNA. But first, it hits the start codon, which is AUG. Once it realizes the start codon, it would then bring the next one over into the E site, like this. It would transfer this over, like so, and this one would leave. And then the next one would show up, and it would keep going and going and going until a long strain has been made. We've reached our stop codon, which is this. It's made out of protein. It goes into the A site, and then it will not let any more through. And then it would move over like this. This is termination. And then it would break off. This is called hydrolysis. And now we have our polypeptide chain. Now that we have our polypeptide chain, which is just
Whoa, JJ. What made it stop?
That's a good question. The thing that made those stop was one of the three stop codons. UAG, UAA, and UGA. Isn't science a great thing? Now then, before I was rudely interrupted by my friend, this is a polypeptide chain, which is just a really fancy way of saying a bunch of amino acids held together by a peptide bond. This is now going to be sent into the endoplasmic reticulum. See you there. Now that our polypeptide chain is ready, it can then be transferred into the endoplasmic reticulum. And then it will go into a vesicle, which then, after it goes into the vesicle, it will transfer into the Golgi body. There, it will be processed and fold it up so it could be ready. This is protein. Then it will be shipped outside of the cell. It's our episode on protein synthesis. See you next time, fellow scientists. Have a good day.
Bill Nye the Science Guy.
Perfect. Now that we took off all the introns, this thing is ready to go to out of the nucleus. Hey Bill, catch. Good catch. And now we have our thing.